Hello, everyone, and welcome to today's webinar, Rare Disease Clinical Trials, an Opportunity for Differentiated Services. My name is Colleen Lezovitz, and I'm a Senior Marketing Manager at Worldwide Clinical Trials. Before we kick things off, I wanted to go over a few things. All attendees are on mute, but please use the questions window to submit any questions at any time pertaining to the presentation, because we will be reserving time for a Q&A session at the end. This webinar is being recorded, and all attendees will receive an email with a link to access the on-demand, so keep an eye out for that, most likely tomorrow. At this time, I would like to hand things over to Worldwide's Chief Medical and Scientific Officer, Dr. Michael Murphy.
Well, good morning, everyone. My name is Mike Murphy. I'm Chief Medical and Scientific Officer for the company. My particular area of interest is translational research, but also includes generating data and programs that would resonate with other stakeholders other than the FDA. And I'm joined today by my good colleague, Leslie Weather. I'll introduce herself in 1 second. So the agenda today is kind of a neat one. We thought we'd engage two different but complementary themes. One is kind of redefining the method and kind of an exploration of the opportunities afforded by orphan research and development to push envelopes in terms of program and trial design. And then the other side of the street, very complementary, is redefining the art. And Leslie will be speaking to us about methods of patient engagement, patient retention, with some very unique operational ideas that are only really applicable to orphan disease research. Well, we begin with the poll question. This is usually very helpful to help calibrate the audience. So let's see what the interests are in the background. So if you could, Colleen, please. So how many of you are engaged in discovery or development research in an orphan disease indication? Notice we include both discovery and development in the question. We're asking you to please select only one of these. So you are actively engaged in one or both. You are not currently engaged, but your organization is considering this. Or you're not engaged in either of these activities, but you're very interested in the topic. So we're not going to vote, but if you could please select one. Okay. Okay, fantastic. Well, we have a diverse audience. Leslie, we have our work cut out for us a bit. We hope that we've calibrated our presentation appropriately, to, given the mix that's in the audience. Now, the first issue is, so what exactly are we talking about in terms of orphan diseases? Of course, many of us on the phone will know that. This is defined by prevalence figures, which differ between the United States and Europe. Occasionally, people will add the ultra designation, which adds an additional dimension to the story. It turns out there are over 16 different terms that people have used with the association of ultra, but the more common definition is that derived by NICE. What does it mean? It means two things: difficult to find and difficult to treat patients. And somewhat more interestingly, the opportunity for innovative trial and program designs. Now, phenotypic variation is common. Frequently, multiple organ systems are affected. If you just look at the left-hand side of this slide I put together, the first 5 or 6 bullets show endocrine disorders, neuromuscular disorders, CNS disorders, dermatological conditions, respiratory disorders. They're all over the place. It is true that cancer is mostly dominant, with about 36% of all orphan indications. But I found it remarkable that the next highest category was simply called miscellaneous. So lots of different targets out there, coordinated multidisciplinary care, which greatly complicates trial design and execution. Lots of transition points. I call them transition points in clinical care, particularly for those diseases which begin in childhood and infancy and transition into adulthood. Over 7,000 diseases, technically, affecting everyone worldwide. So lots of opportunity for very differentiated methods, and it is a wonderful time for us in this particular arena. We have great R&D and commercial incentives, 10-year market exclusivity in the EU as an example, a very favorable regulatory climate for both novel and repurposed products, an opportunity for innovative, efficient study designs, in which is going to be the focus of my presentation. Many diverse measures, not only diverse in terms of the assessments that are going to be employed, but also diverse in the sense of who is performing the measure and where the measure is actually completed. Some very unique operational solutions, as Leslie will point out in a minute. Perhaps we monitor and organize study conduct differently here than we do for more traditional indications. Really like this participatory research models. Now there are lots of webinars and seminars on this topic. Frequently they focus on patient accrual and retention. I would like to focus on this particular aspect in terms of site relationships, and also including the family in the design of the study, which inherently will take care of accrual and retention. So very participatory, and certainly the globalization in clinical research. It's difficult to imagine executing these programs without having an interesting international footprint. So a very exciting time for us. From a design point of view, look at the smorgasbord of applications on that left-hand side. If you're a clinical trialist, you have to be drawn into this area. Now, most of us read about these designs, but infrequently have an opportunity to use them all. The orphan drug space actually allows you to legitimately consider some of these as part of the program composition. Mostly when you look at the literatures out there, the types of designs cluster into two domains. One relates to minimizing sample size, and the second relates to maximizing treatment. Minimizing sample size, of course, is really quite an interesting approach with various adaptive randomization schemes, longer trials, more events per patient. Those of you that joined us a few weeks ago in the heart failure program, we talked about recurrent hospitalizations as a way to minimize the sample size in those studies. Using high-risk patients or those with genetic testing, leveraging the sample, risk stratification. Using continuous or repeated measures, or borrowing from my colleagues in cardiovascular outcome studies, composite endpoints, which are yet another general way to reduce sample size burden. Using relaxed alphas. Not every study needs to, as I like to say, worship at the shrine of the p-value. It is entirely appropriate, depending on the program, to have a more relaxed approach to hypothesis testing. And if there was any space that demands more attention to Bayesian frameworks, we must agree that it is the orphan indications that would lend itself to that quite a bit. N of 1 designs and balanced randomization, crossovers, and lots of permutations, different ways of organizing data and developing evidentiary standards of safety and efficacy, all a part of the mix that can occur in this space. In addition to observational research, we have lots to look at here. Many arrows in the quiver, from cross-sectional studies through retrospective and prospective cohort designs, nested case control studies, to arguably the gold standard. Notice I said arguably the gold standard, randomized controlled trials, self-controlled observational designs. All of it is good. This entire continuum goes from hypothesis generation through formal hypothesis testing. And rather than either or, one central theme for today's presentation is that it's a mixture of both. Observational studies complement and serve the interventional program that we all wish to complete. And types of questions are really quite neat. We can inform a protocol design through observational studies. Frankly, some companies are using these to complement a submission strategy. Isn't that interesting? Certainly, we want to use this type of research to shape formulary and reimbursement decisions, and they have many, many different objectives: natural history of disease, burden of illness, treatment pathways. Really an exciting area, treatment pathways. How did you arrive at the point of using my product? How did you get here? What medication did you use? Who treated you? Who ended diagnosis? Post-marketing usage, on and off label, and of course comparative effectiveness. Now that little avatar holding that Swiss cheese product development plan on the left-hand side is simply to highlight what many of us have encountered, is that it's basically our reaction to what had been a strategic program of development to only realize that we did not quite have the portfolio of studies that we originally thought would have been useful, a lot of gaps in coverage. So a mixture of interventional studies of variety of designs and a mixture of observational research would work very well in this particular space. Now, you know, the old story is if you've seen one orphan indication, you've kind of seen one orphan indication. This is just a brief way to illustrate all the different things that people encounter depending on the indication you're working in. You know, for example, we have the behavioral variant study we just recently completed, where the medical monitor really became an extension of the sponsor and a representative of their program. It was critical to site engagement. A Gaucher disease program, if there was ever a place where the design and operations need to speak to one another, this is one. Quite difficult to fly patients across 11 time zones from Vladivostok to Moscow when there's a very tightly meshed visit structure to accommodate. Or pulmonary arterial hypertension, a few companies are involved with that. That's our problem. Everyone wants the same patient, and there are only very few centers actually capable of doing it. Hemophagocytic lymphohistiocytosis, you know, we're familiar with this. Multinational studies, multilingual and complex informed consents. And finally, I have to add this one on the bottom left here, thrombotic microangiopathy, you know, because sometimes there is rare and then there's really rare with individuals where the prevalence is low and the presentation for study is actually, you know, very difficult. The idea of this slide is, and at least from our experience across over 56 studies, is we borrow from Peter to Paul. We understand the dynamics of every orphan disease can be somewhat different, but there are certain commonalities and certain lessons learned that we can incorporate in design and in the operation of these studies. Now, we want to define the box, but we also want to step out of the box. So in general, we have I call the conundrum of placebo. No family, no patients wants randomization to placebo, our standard of care, quite understandably. What is the contribution of historical data on a group level, for example, through a registry, or on a patient level? Is that informative in some respects, both to program design, if not even registration? I mentioned that there's a multiplicity of effects in most of these diseases. What is the most sensitive and the most specific measure in a multi-organ disease state? And actually, who's completing that? Is that the physician, the family, the patient, and where is it done? A few weeks ago, we were privileged to give a seminar on siteless clinical trials, moving the assessments out to the family and not so much into the clinic where it's appropriate. When a patient is traveling 1,000 miles to attend a clinic for a visit, that is something to be considered. So innovative design, this is an opportunity for really out-of-the-box types of studies, hybrid monitoring strategies that Leslie will speak to. And then another one that's come up recently, I call it kind of a one-and-done registration strategy. Is that tenable within the framework of current regulatory guidance? I mean, how much can one actually push the envelope in terms of program design? Now, here's a neat article. Those of you in orphan space probably have read it. Those that haven't, I do recommend taking a look at this. So this is from Sastanowski's article, and it really stimulated a discussion of what is possible. An overarching review of flexibility in evidentiary standards, at least as expressed by the FDA. Now, all of us are familiar with the upper right-hand corner there, which is the conventional approval process. You know, 2 adequate, well-controlled studies, each meeting a pre-specified primary endpoint, p-value of less than or equal to 0.050. Notice I used 3 decimal points. We're all familiar with that. But there are also some other opportunities that were brought up in this particular review. The administrative review, 2 different methods which affect either the number of studies, maybe a single really excellent study, or the type of evidence that might be required, so-called Subpart H approval. What was really, and of course there's always case by case, which is always an exception. What was really fascinating about this review is that 2/3 of these 27 non-cancer, you could imagine in oncology this might be true, but there are 27 studies not related to oncology but 2/3 of them were approved based upon some evidence of flexibility on the part of the agency, who actually is quite accommodating and recognizes the need for early patient access. I think this one slide says it all to me. In terms of a registration program, an opportunity to think through the strategy of the approval seems to be a wonderful in this particular case. Now, what would be the framework? Well, we're all familiar with accelerated, fast track, and breakthrough designations. The breakthrough designations particularly are so attractive. There are, not exclusively in oncology, by the way, there are about half of them that have been given this designation are in targets which do not involve oncology agents. Other sentiments are buried in the literature, such as there's no specific minimum number of patients, or another one that says fewer, smaller, shorter clinical trials. And then more recently, the one that captured everyone's attention earlier this year in Duchenne's, the use of external controls, such as historically controlled trials. How exciting is that? An ability to use the data that's already available to support the registration of a compound. In that setting, and against that backdrop, could you not explore the following? Given that your primary outcome is significant, that your secondary measures are supportive and convergence with the primary, and that good GCP compliance is assured, given that that is true, is it also true that you already have what I will term an informative study in the bag that one could reference to support the approval based on one study? Is it possible, it's just a question, that a registry with case control matching, for example, could contribute to the evidence of efficacy? There's a really very interesting article in Gaucher's disease that kind of attests to that. Is it possible that prior clinical data with a post-marketing commitment to perform a registry, as an example, is it possible that that would be, with everything else, sufficient in order to accelerate the approval of this compound? I think those are exciting possibilities for all of us. Now, but people have reminded, Mike, sometimes it isn't always true that one is sufficient. So this recent publication came out, many of you have probably seen it. There are 22 case studies here where the phase 2 data and the phase 3 trials had divergent results. Ouch. 14 out of 22 failed to demonstrate efficacy in phase 3. 1 out of 22 failed to demonstrate safety in phase 3, which is really remarkable. And 7 out of 22 failed to demonstrate either efficacy or safety in phase 3. So what are the issues in here? Little caveat to that previous slide that I showed. Well, the common one, the proposed surrogate endpoint that people try to develop doesn't equal a clinical endpoint. We're all familiar with the CAST study, the kind of the emblematic of that particular problem. Cardiac arrhythmia suppression trial done years ago. Unfortunately, all too common, that is true. Occasionally, there are no surprises here. I put this no surprise. The population of phase 3 was just remarkably heterogeneous, and therefore there were no differences there. That commonly occurs in many different indications. Possibly predictive, therapies were dangerous in combinations. The CYP450, for example, data that you had, and the limited trial data that you have didn't point out combinations which would have been deleterious when used in a more representative sample. One that we all know is true, that patients' populations drift. As the programs evolve, we broaden eligibility criteria. We attempt to make the data more generalizable. But in these particular cases, both risk and response often look quite different. And finally, I'm reminded by when I read this review of the so-called Will Rogers effect that commonly occurs for first-in-human studies. Will Rogers, I never met a drug I didn't like. So very commonly encountered in early phase research, not confined to any one therapeutic area or type of compound. And indeed, when you look at this review, they're not therapeutically area specific. Divergent results can occur across all indications. They're not related to mechanism of action data specifically. They occur across all classes of investigational product. And what does it mean for us? It means that when we attempt to truncate a clinical program, even with possibly breakthrough therapy, with all the caveats I've just mentioned, it's entirely possible that the limited data we generate will generate a limited utility of any benefit and risk. We will misjudge the benefit to the risk. This means that the development of the compound maybe should extend into the post-marketing authorization period. We need to gather more data and maybe different types of data for some of these different agents. This would be particularly true for the ultra-rare disorders where you actually couldn't conduct a traditional program. So the rise of registries, I love the title. So characterized by representative populations, representative providers, we can examine so many different types of things, such as interaction effects. By that I mean, how does your treatment interact and either augment or diminish the effect of other types of therapy? We certainly have a much more heterogeneous patient group, all of whom have very different disease trajectories. And they come in different forms, as we know, patient self-reported, professional reported, or a combination of patient self-reports and professional reports. Everyone's interested in this. There are lysosomal storage disorders, for example, Fabry's disease, Gaucher's, have had the longest tradition. There are at least 5 different registries there. But other disorders have also incorporated them. There are at least 15 other indications that are non-lysosomal storage disorders that are also using this particular approach. They all have limitations which have been recently reviewed. But how do you use it? Well, you can use it to inform safety, which is the traditional way that we would think about it. We could also use it to inform value. You know, how does it fit into the therapeutic armamentarium, into the mosaic of other treatments that might be available for these patients? What is the impact on healthcare utilization? That's another objective. Or how does it inform future trials? I think that might be the most interesting part of this. What can it tell us about disease trajectory and presentation that will allow us to use some of those innovative designs that I previously mentioned? And then somewhat more provocatively, how can it be used to support registration? We have some precedent for that. Is it not possible that this could be a companion initiative with the interventional research that we've done to support registration? So let's take a look at this in terms of a poll question and see how people think about it. So Colleen, if you could, please. Okay. Now, I'm going to lead us through this one. So for clinical research within an orphan indication, this is going to be a hard one because you can only pick one. Which do you consider the most important program element? Now, the first choice is what I call data mix. That is the blend of observational and interventional studies that you may produce. That's interesting. What about, I call it process, kind of a regulatory framework and flow? You know, the accelerated approval, breakthrough therapy idea. Would you rank that as number 1? And then third on the list, trial organization. Is a dedicated, competent, engaged CRO project team the most important? So just pick one, please, and we'll take a look at the results.
Like about 30% of people have voted, so we'll just wait a few more seconds.
Well, we knew this one would be difficult, Colleen, because there are very different constructs presented here. Okay, well, I can see the uncertainty in the mix here. Although half of you felt that a coherent, accelerated, perhaps breakthrough therapy approach vis-a-vis regulatory affairs would be the most important issue. But data mix and trial organization certainly look interesting, and this is where the art of clinical research comes in. So my good colleague Leslie now is going to take us through those particular considerations. Leslie, if you could.
Good morning, good afternoon, everyone. Thank you, Dr. Murphy. I'm excited about the opportunity to present today. So my name is Leslie Wetherill. I'm the Rare Disease Franchise Lead at Worldwide, and today I'll be talking about engaging the stakeholders and the study team, as well as ensuring data quality. So Colleen is going to start us off with a poll question.
Okay, 1 second. I'm going to go ahead and share that out. So this poll question asks, has your organization engaged with patient advocacy groups for rare disease trials? Okay, almost half have voted, so I'll wait a few more seconds. Okay, I'm going to go ahead and share those results.
Okay, great. So we have about half of the audience has engaged with patient advocacy groups, 21% have not, and 31 are considering. So hopefully I'll convince you this is a good idea for those on the in the consideration area. Okay. I'm trying to advance the slide. Okay, here we go. All right, so building off what Dr. Murphy has stated, the protocol design needs to be carefully considered, but the additional step is engaging the patient and the physician for feedback on the illness. So when we talk about treating illness versus disease, we have to consider how the illness affects the patient lifestyle. So an example would be, patients with a coagulation disorder. Those patients are used to self-treating when they experience a bleed, and most have been self-infusing since their preteen years. But if a protocol was written where the physician administers the investigational product, we might put unnecessary stress and inconvenience on that patient. Another example is bullous pemphigoid disease, which we're working on with our colleagues at Immune Pharma. So they've worked closely with the patient advocacy groups for patient awareness, and they've even sponsored patient travel for Rare Disease Day for Rare Disease Day conferences. And they've incorporated a patient-reported outcome measure called the BPDAI, which is an activity index, and that's unique to the concerns of the patient population. It's aimed at aligning the response to the specific condition for bullous pemphigoid disease. So you can see that patient advocacy groups are a great resource for us. They provide comprehensive feedback regarding the illness, and just a reminder that we have to take this flexible approach. We have to consider that feedback and make sure the protocol is manageable and fits into the patient and caregiver lifestyle, and that ultimately we're providing benefit to their quality of life. Okay, so rare diseases often manifest themselves during childhood, and the patient advocacy groups and the families are usually well educated in the treatment and the disease. So in fact, some diseases have a genetic substrate that informs drug discovery. Natural history isn't always well known for these patients, and a companion program can inform the disease trajectory. This provides the ability for us to map out how the patient presents to a program, and it amplifies what Dr. Murphy said earlier about commitment to the patient. We have to plan proactively for their long-term commitment to the patient. We should expect that a close interaction with all the stakeholders all the way through the process, and that includes the families, the doctors, the patients, the advocacy group, and the sponsor. And we have to maintain these relationships and move all these stakeholders from phase 1 into post-approval stages. Okay. So you might be thinking, how am I going to engage with all these stakeholders, a lot of people to consider in these kinds of diseases. So when we're talking about engaging with physicians and sites and clinics, we can work with them on their patient databases and identify the patients that they may have been treating in the past or are currently treating. And we can also provide links to social and digital assets for them to use as referrals. We can give resources to provide information to their network of colleagues to help them identify any patients they might be seeing. When we talk about the advocacy groups, they can list the clinical trial on their advocacy website, and they can also connect via social media, usually Facebook. There's also blogs and forums that patients may use, and we can provide information on blogs and forums to reach the caregivers and the patients. So we don't know how well social media really works. Internally, we have some mixed reviews, and Dr. Murphy, feel free to jump in here, but I'd be curious to hear of any experience on the phone. Do hits on social media translate to enrollment? That's difficult for us to measure usually, but the exposure to the clinical trial information is good. Once the patient is enrolled, patients may complain about an assessment or their treatment, so that may be something that we can't control and is not necessarily favorable for us. So I don't know if you have anything to add, Dr. Murphy.
Well, that's exactly the concern that Leslie and I have frequently talked about, and we don't have enough experience with it to come down definitively on a recommendation, except to say it's a powerful tool, and with any powerful tool, it can cut both ways. So that's an interesting additional, type of technology that lends itself to use here.
Okay, great, thank you. Okay, so now that we've engaged the physicians and the community, we have to assemble a team that can build on that foundation. So as Dr. Murphy sometimes says, this type of research is not for the weak of heart. CRAs will not have experience in the 7,000 rare diseases out there, nor will any of us. So we need to understand that there's a different approach to rare diseases, and we need to be able to quickly adapt to that indication and the style of the sites that we're working with. So we need a confident and experienced team in doing so. They need to be able to provide good clinical practice support. They have to understand the disease and the pathway of the patient's care, so that they know when will the patient present themselves in order to enroll into the study. So we need to be able to quickly learn that new indication and understand a different approach in the rare disease. Our medical monitors will be actively engaged, and they need to understand the illness not only to support and train our team, but to act as an extension of the sponsor in the field. The team will need to be able to speak about the protocol to the key opinion leaders, and these people are experts in their field. We can never match their expertise, but a common understanding is needed to be a good partner to them. If we can understand the pathway of the patient diagnosis and the care, then we can follow that to ensure that we're providing materials to the patient at the right time. The entire team must ensure this engagement ethos so that we can be successful. Okay. Sorry, my slides are advancing slowly. Okay, now site engagement is a key area of focus for a number of reasons. Due to the low prevalence of clinical trials in certain indications, some of the sites that we reach out to may be new to conducting clinical trial research. They may be very well versed in clinical research, but in these types of trials, they may need our support. So we will need to be there to train them not only on good clinical practice, but in IRB submissions and updates and SUSAR reporting and many other elements that may be new to them. So the CRA will need to pay close attention and guide the site as needed through that intense process, leading up to their activation as well as the support once they start screening patients. So these aren't studies that we normally monitor with a set frequency, like every 8 weeks, where we're seeing the sites, quite a bit face-to-face. This is a more streamlined approach that's focused on individual patient activity. So we may not go out to the site until they start screening or enrolling a patient. That allows us to support the sites at the right time, and can help us not to create unnecessary cost and visits to the site. So a remote interaction with the site is needed to be able to support the site through all these questions regarding the protocol and the nuances of treatment. That interaction may be infrequent because they might screen a high number of patients only to recruit one, or they may know of only a few patients to reach out with a condition, and we may know that well ahead of when they start screening. The CRA can help the site to make sure that they have the right messaging and the right patient communication tools to convince the patient to come in and discuss the opportunity, and then help them maintain the patient interest in the participation on an ongoing basis. Okay, the next area I wanted to talk about was quality planning and discipline. So due to the probability of the low number of patients per site, the sites have little opportunity to learn from any mistakes or misunderstandings of the protocol early on. So the attention to the data quality is elevated for the entire clinical operations team, meaning data management, statistics, medical writers, etc. These additional steps that are needed to ensure that the output of the trial is consistent and reproducible. So the first step is for us to maintain a proper medical oversight. And as I said before, the medical monitor can be an extension of the sponsor in the field in site engagement, but it also includes reviewing eligibility, especially if patients are they have a long history of their disease. It may be difficult for us to navigate through all of the charts in order to make sure that they are truly eligible for the trial. We can also provide lab alerts that help point out issues with safety or efficacy to the medical monitor to ensure prompt reporting of events, or for them to reach out to the site if they're having any kind of issues with the patient. We recommend that the CRA be on site the first day of dosing for that first patient. And that is just there to provide an extra layer of support to the site, so that if they have any protocol questions or any misunderstandings about any of the equipment or anything on site, that we're there to support. Our data management team can help support us with listings, with key performance indicators that allow the team to see trends across sites. It also helps identify the need for further site training. So if we can see that a particular form or data collection point is generating a lot of queries, then we can see that that might be an area we need to go back and make sure the sites really understand how to collect the certain piece of data. It also can help us determine if a site is falling behind in their data cleanliness, and then we can increase monitoring as required to get them quickly back on track. The follow-through by the team ensures that we avoid replacing patients in trials with a low number of patients contributing to the overall data. And in summary, proactive planning for data quality is important to our successful delivery of a project. So now I'll hand it back to Dr. Murphy for some final thoughts.
Well, very good, Leslie, and very interesting discussion regarding how to operationalize this study. I was fascinated to hear some of your comments here, because they are a little bit different from traditional indications. Now, I thought about, so how should we summarize this over the presentation for the last 40 minutes? I thought there were three things that really capture it all. The first is engagement. So this is sponsor CRO integration that transcends a transactional relationship. Transactional relationships were characteristic of sponsor CRO engagements from the inception of this industry in the 1980s. But for the last 10 years, really, I think that science is so sophisticated, the clinical targets are so nuanced, that an ability to work together and learn from each other in a strategic program rather than a transactional relationship probably is ideally suited to the orphan disease space. The second one, which is near and dear, is creativity. I call it, one eye on heaven with both feet on the ground. Now, if you're a clinical trialist, you have to love this particular area. Observational studies, which there are many, a whole panoply of interventional designs that would be at least exciting to consider. Notice, didn't recommend them all, but the ability to sit around the table and think through all the different permutations that might characterize an efficient and reliable clinical program would be very exciting. Cognizant of the regulatory frameworks, which I think are very permissive currently, Family and patient sentiments, which are everything. Everything in design goes back to the family. If you build it in the design, it'll be there when you finish the study. And the limitations we must be aware of, which are inherited by producing what really would be a small amount of data upon which societal risk have been based. Just as I showed you with the 22 studies that looked terrific in phase 2 but failed for various reasons in phase 3. So creativity is a second component. And finally, the most important one is commitment. The way I always say, a sword in one hand and a pharmacopoeia in the other. That is, you have uncompromising commitment. It is not calibrated. That expression we use around here to say that we stand with the patient, we speak for the sponsor, we want to give these innovative therapeutics the fair shot at showing value, and under good circumstances, we would like to contribute to successful product registration to assure success. So those are our kind of thoughts. Now we have opportunity today for a number of questions. We usually get quite a few. And while we're waiting to receive them, Colleen, I'll just start out with one that I am frequently asked. You might find it interesting. So it's kind of like this, you know, from a clinical development within an orphan indication, you might say it doesn't have the scale, doesn't have the patient numbers and so forth. And so the revenue stream for a CRO must be very limited in contrast to more traditional targets. Why would there be such an interest? Well, there are three things. One is opportunity. You know, everyone seems to be orphanizing indications is the way I commonly refer to it, even those for diseases which have not historically been identified as such. So the number of opportunities is certainly there. There's a great deal of opportunity for innovation and strategic discussion around these programs. The art of the trialist, the ability to integrate experimental design with impeccable operations. It doesn't get any better than this particular space. And finally, the notion of having impact. You know, the unmet clinical needs here are so enormous that to contribute to the registration of a product for an orphan disease, I can't think of anything else that would be more rewarding. So our commitment, for example, has been predicated on that. We frankly did not have a strategic objective when we started out 8 years ago doing this, but we kind of fell into it because it's the natural affinity of the raison d'etre of our organization, which is an ability to integrate, as you see on this slide, scientifically minded and medically driven with operational acumen. Do we have any other questions, Colleen? Let's see if we have a few coming in here. I have one for you. This came in, Leslie, if you don't mind. So what type of key performance indicators or key quality indicators do you suggest for site assessment of quality?
Okay, so usually any of the, we have a set of standard key performance indicators that we use for most trials, but the ones that are really focused on the site level detail can give us an idea of how a site is performing from a metric standpoint. So that can allow the team to focus in on where we have the most problems, like if we have particular sites that really need an extra effort in monitoring. So I would look at protocol deviations per site versus, how many subjects are enrolled at that site, how many open SAEs they have versus the total SAEs reported, and that would indicate, that they're able to quickly close those things out. And if things are remaining open for a long time, it can give us an indication that they're struggling to understand, what we need from them. Pages entered versus SDV'd or query number of queries per patient just gives us an idea of, are they able to keep up with the data flow, and if they're getting behind, indicates that we should go back out and monitor as quickly as possible.
So here's a related one, Leslie, I think kind of has to do with this. What kind of training do you suggest for the CRA staff working on a rare disease trial? How do you see that?
Yeah, so the in-depth training that the medical monitor would provide on the indication is really important because we really need them to understand the disease. And for the most part, our CRAs are not going to have experience in these ultra-rare conditions, and so that really needs an in-depth training on the medical side. But also they need to understand that there's a different approach to rare disease trials, and that the level of site support and the level of support in patient enrollment, patient retention, and things like this is it's much more elevated in these kinds of trials. So they need to understand that they need to support the site in those aspects and make sure that the site understands how important that is to us. So,
Leslie, do you think, by the way, that we need a more seasoned field monitor? You know, the pedigree of the individuals that we're sending out for these studies. Do you think it's different here than it might be for some other traditional indications?
Yeah, I think definitely the years of monitoring experience and their experience with good clinical practice is going to be really important, so they can help train some of the sites that are a little bit newer to this than others. So they can really be an extension of the site and provide, you know, phone support or on-site support as necessary. But you know, if they have a medical background at all or a nursing background, you know, sometimes that's very helpful. Because it's a little bit easier for them to understand the nuances of the patient pathway and things like that.
Mm-hmm. Well, here's one for me, I think, is how do we overcome lasagna's rule of patient recruitment? Now, everyone that's been in this vineyard for long enough knows this rule. That is, the number of available patients decreases exponentially as the date of trial launch approaches. Everyone has lots of patients until you actually want to start the study. And Les, I know you have ideas too, but I'll begin. I say it begins with the design, so that I'm maximizing the type and amount of data I can get from every single patient, thereby reducing the overall sample size required for any given patient. But the second has to do, Leslie, with kind of reviewing the database that might be available. There are only a few centers mostly that can do these studies. And as we know, these physicians are also managing the patient's illness. So how do you do it, Leslie? When you do a site qualification visit and you want to bring us, how do you do that?
Yeah, so what I recommend usually is that when we start with the feasibility questionnaire, that feasibility questionnaire is much more detailed than, you know, our typical feasibility questionnaire. So we want to know how many patients they're currently treating with the disease, and then we can, sometimes those numbers don't make sense. We can go back and ask, are you really seeing 10 patients with this disease, or, is it, 10 patients that you've ever treated with this disease? So, we really have to go back and explore the answers to those questions when we go to do the pre-study visit at the site. And we can help them look through, if they allow us, we can help them look through charts and things like that to see if these patients really qualify, or if they understand, that we're looking for, maybe a subpopulation of a greater population. An example would be like a cancer trial where, we're looking for a specific biomarker or a specific subtype of a type of cancer. So they may see plenty of breast cancer patients, but are they seeing HER2 positive breast cancer patients in their third line of treatment? That's a much different patient to find. So just really making sure that they understand what we're looking for, and then helping them identify how many patients they currently see in their database. And then what I ask my team to do is just to keep track of those very early pre-screened patients. Although we can't collect any information, to really identify those patients at that point in time, we can say, okay, they have 2 patients that they say that they are treating, and then when we go back to do, the initiation visit, etc., then we can ask about those 2 patients, and ask them to look into where they are in their treatment, and would they be willing to come in and talk about the trial at that point.
Yeah, the other thing we do there, Leslie, is those screen failure patients, we've been recommending this frequently now, enroll them into a concurrent longitudinal cohort study so we can start developing some information about disease trajectories and standards of care, which we can use in other types of analyses. So every screen failure patient is simply a patient for another type of study. And in regard to knowing the patient, Leslie, if that site doesn't know them by their first name, I get a bit anxious with them. Okay. So here's another one, Leslie. How do you select which patient advocacy groups to work with? Well, I'll take a crack, but I know you have ideas on that one too. For me, it's very simple. Well, there's 7,000 diseases, and the odds of us actually knowing the disease and illness here is pretty small for any one of them. And the way it would work for us on the side of the street of the design side is we can read all the literature that's out there, and it's quite interesting, and there's really beautiful literature, but you know, that doesn't mean we understand the illness. So the first phone call I usually make is to a group that is a patient advocacy group, and what I really want to hear is tell me about your son. Well, you know, Dr. Murphy, if you schedule a fasting lab on the same morning I'm doing a 6-minute walk test in Duchenne's, that's not going to work out so well for you. I want to understand the illness, not the disease. And a patient advocacy group that is willing to engage you on that level before the protocol is designed is invaluable. Leslie, what's been your experience there?
Yeah, I would say that, you know, there are some web links where you can find quite a bit of information about advocacy groups. So one example is rarediseases.org, which is the NORD website. And if you type in the rare disease that you're looking for in that, they have like an information link, it will pull up some information about the disease, and usually at the bottom of that it provides all the patient advocacy groups that they're aware of. Now some very, you know, some have a large number of patient advocacy groups available. Others may not have any. It may just list something like March of Dimes or something along those lines. So you'll see that for different diseases there's a different level of advocacy groups available. Now, which ones should you reach out to in that? If 5 are available, I don't think there's any harm in reaching out to them all and trying to get a feel for, you know, can they help you in clinical research support? Would they be able to advertise on their web link? You know, can they work with us with that kind of information? Or is it structured a little bit differently, and that's not something that they would necessarily be open to? So it's worth a phone call and just discussing the trial with them to see, you know, what kind of support they can provide to us.
Here's another great one. This is probably the most common question we get is how do we develop a reliable, meaning predictable, patient enrollment projection when we engage for orphan disease services? What are your thoughts on that one, Leslie?
Yeah, so this is a tricky one. So we don't normally see the traditional enrollment graph, you know, that we typically present in the early stages of a trial. It's usually more dependent on the site, you know, like a per site type of commitment. And so we can put together an enrollment graph based on that kind of information that we're getting, which is much more manual than the typical, you know, we're expecting a 0.25 enrollment rate. Usually in these kinds of studies, we're predicting something like in a 0.1 or even less than that in some cases. So it's really how many patients per site. So if that site's saying 1 to 2 patients as an example, if we put in a 0.1 enrollment rate, we would need at least, you know, 12 to, you know, or 15 months or so for it to actually calculate out patients. So it's not very helpful in that way. But if we can do more of a manual, you know, manual type of graph based on the information we receive from the site, and just keep in mind that not every site is going to be able to, you know, provide a patient. In some cases, we're going to have to open more sites than we think we probably need, because if that site only has, let's say, 2 patients that they can reach out to, and those 2 patients aren't interested in clinical trials, you know, we won't know that until after they're able to reach out once they're activated. So it can become very difficult. And
yes.
I'm sorry, someone have a question?
Yeah, so there's another one here that I thought was an excellent one, Leslie. So how do you get your trial to be top of mind for a PI at an academic medical teaching institution when his or her daily focus is necessarily patient care and not your study? Well, I'll take a crack, Leslie.
Okay.
It begins with the design. You know, the design has to be compatible with standards of care. If it's discordant with the care that the patient is routinely administering, already we start out with a problem from that PI's perspective, and indeed it should be. So the design has to be compatible with ongoing standards of care, just to begin with. Secondly, the engagement needs to be multidisciplinarian. Everyone on the team, from the physician through the project manager to the CRA, is there to contribute to what I call part of the solution, which is how can I make your day-to-day easier in the context of doing this study. Our experience, Leslie, has been, and you know this, that for many of our orphan disease indications, the PI has actually never done an interventional study, almost by definition. And so the site needs more attention rather than less attention to the details of running a study. So by doing that consistently for the study where you have been assigned, it raises the visibility of the company. It raises the visibility of the importance of the question we're attempting to ask. And I think that's an excellent question. And a tendency that we've seen recently, this is not exclusively in orphan disease research, but it is very common for early phase investigation, is for sponsors to increasingly go to fewer centers, but better centers. I say fewer, better centers. And so developing that relationship is really an important part of working in this space. You have any other thoughts on that, Leslie?
No, I think you covered it for the most part, Dr. Murphy. I think that, we have to keep reminding the site when we're talking to them, we're not spending as much time on.
Leslie, we may have lost you there. So we have one more, everyone, which is
and others on the involved in the trial to remind them, to look for patients, ask them if they've reached out to any patients they're aware of, and then give them some materials to reach out to the patient.
Yes.
And remind them that we understand it might take a while to get the patient to come in and discuss the opportunity, so just be patient in that, but then try to keep the trial top of mind by reaching out regularly and checking in with them.
Yeah. The other thing that's great here, Leslie, is I like to go out and ask them about the assessments, because sometimes the assessments are a little arcane. They're unique to that particular indication. You don't have much experience. I'd like to talk to the study coordinator, say, what's it like to do that measure in your protocol? And usually the comment is something like, I don't know who put that in the study, Dr. Murphy, but boy, is that causing some issues for us. Or maybe, this is really a neat assessment. It seems to be addressing the needs as it was originally intended. So the back and forth of it before you design the study and the back and forth during the study, I think is key. Leslie, time for one more question, then we'll have to stop. Are clinical trials in children different than in adults in terms of rare disease clinical trial design? How do you see that one?
Okay, well, obviously with the children, the parents and the caregivers have a very important voice in the design of the trial. We need to make sure that they feel comfortable with the design, that they feel like that we're not putting too much undue stress or too frequent of visits or things like that for the children, so that they can attend school. We're not pulling them out of their normal day-to-day activities, due to the trial design. So it's really trying to make sure that we are being adaptive and flexible in our approach, so that, like, let's say if they need to do lab draws, if they can do lab draws like in the home health care setting, or if they could do that at a local clinic versus going back to the university hospital, makes it much more convenient for the caregiver and the patient And so, you just have to take all those things into consideration. There are many other things to take into consideration with pediatric trials. I don't think we have time to go into that, but maybe that's a topic for a future webinar.
Well, everyone, we've reached the hour of our webinar. We really appreciate your attention and courtesies. We look forward to speaking with you on the road. So thanks, everyone, and on behalf of WCT, we wish you well.
All right.
Thank you, Dr. Murphy. Thank you, Leslie. So this concludes our webinar. Thank you for joining, and we hope you found it informative. If you did ask a question that we did not get to, we will reach out to you individually. And as I mentioned earlier, we have recorded this webinar, and all attendees will receive a link to the on-demand, most likely tomorrow, so keep an eye out for an email. And for more information about Worldwide, we invite you to visit worldwide.com. We hope you enjoyed and have a wonderful rest of the day.
Thank you so much.